hospital acquired exercise is 20 marks, and viva was of 30 marks. Remember, here viva was of 30 marks based on the paper 1 and paper 2. 15 marks each. Now, paper 1 will cover the general malarial system, respiratory system, gastrointestinal tract infection, and hepatobiliary system. This is the syllabus of paper 1. Viva was 2 will cover the central nervous system infection, musculoskeletal, skin and ST, and miscellaneous topic. So there is only one change in this, where viva was 2 will cover the GIT. It is not in the viva 1. So this is something regarding the practical assessment. Now, the practical assessment of the university exam is similar to the practical assessment conducted in prelim exams. Same. Then day-to-day evaluation we have already discussed previously, so I'm not going into that. But remember, practical record book having 20 marks of weightage. Practical record book, practical journal. I'm talking about practical journal has weightage, and it is complete practical journal, completed and signed practical journal will be a eligibility criteria for the appearance in the exams, both exam prelim as well as in the university exams. I think this is clear. Is it clear? Anybody can give me answer. Is it clear to you regarding the assessment? Anyone can answer a question? Okay, fine. Thank you. So let us move to the first practical. Objective of this practical is describe different types of microscope, its principle and applications. So this is the objective of this practical. So I will run these slides one by one, and we'll try to understand in this objective. Microscopy, use of microscope to magnify the object. So remember, we are magnifying the objects like bacteria, viruses, fungus, and parasites. Now, these objects or these organisms are too small to be visualized with the unaided eye. Now, remember, in microbiology, we are dealing with the diagnosis of the condition, or we are identifying the organisms responsible for the pathogenicity or for the disease. Now, there are two ways to identify the microorganism. One is direct evidences. Direct evidences means we visualize the microorganism under the microscope, or we grow the microorganism in the laboratory conditions, and we identify them by running some biochemical test or by keeping some biochemical test. So these are the direct evidences of the presence of microorganism, or we are identifying them as a causative organism or etiological agent for the disease or for the infection. Second method is to recognize or diagnose the microorganism by some indirect evidences, where either we are not able to see them under the microscope, or we do not have facility to see them, or we are not able to grow them in a laboratory condition. So in this situation, we are diagnosing the infectious condition by using indirect evidences. So let us talk about the direct evidences. Indirect evidences we'll discuss later on, but let us try to understand the direct evidences of the presence of microorganism in the clinical samples. Now, question arises, which microscope to choose? There are varieties of microscopes available, but which microscope to choose? So this is based on the various factors. First is, what is the cost of microscope? What are the features? What is the frequency of use? What is the resolution power of the microscope? And what is the resolution power required? And whether there is so these are the few factors which influences the choice of the microscope. Now let us move to the next slide. Question arises, then what are the applications of the microscope in the microbiology laboratory? Let us try to understand what are the applications of the microscopes in the microbiology lab. Microscopes are used by the pathologists as well, but there are a few differences. Let us try to understand which are the few differences. Preliminary organism identification. I already said, we are discussing the direct evidences or gathering the direct evidences of presence of microorganism in the disease process, or we are labeling this microorganism as an etiological agent. So this is the first use of microscope. We are identifying organism primarily under the microscope. We are also using microscope for the final identification of certain organism. So for the final identification of certain organism, we are using microscope. Say, for example, Mycobacterium leprae. So Mycobacterium leprae is responsible for the disease leprosy. So here we are not able to grow them in the lab. So we are doing a staining of the tissues or staining of the samples, and then we identify them under the microscope. This is one thing. Or preliminary organism identification. For example, if you see a gram-positive organism or gram-negative organism, you see the characteristic or arrangement of the organism, and then you can identify the organism primarily. Then you run few tests and identify them further. Then third use is detection of different organisms present in one specimen. Say, for example, specimen of urine or specimen of stool or specimen of blood comes to the laboratory. So we just see whether organisms are present in the specimen or not. Detection of organism not easily cultivated. I give an example of Mycobacterium leprae, which is not cultivable in the microscope. Evaluation of specimen for the presence of cells. Say, for example, sputum sample or the urine sample, because these two samples, when you try to understand, sputum sample is collected in case of respiratory tract infection, and we want a good specimen of the sputum, which we are expected to come from the lower lobes of the lung or from the lung only, but sample comes through the oral cavity, and there is a possibility of contamination of sample with the saliva. So here we are evaluating this sputum specimen, whether this specimen is contaminated with saliva or not. So we count the number of WBCs and count the number of epithelial cells to label whether this specimen is proper specimen or not, or good specimen or not. Say, similar way, urine sample is collected in case of urinary tract infection, but again urine comes through the urethra, and upper part of the urethra having a genital flora. So we are checking whether urine sample is contaminated with genital flora or not. So here we are evaluating this specimen. These are the two examples. There are other examples as well, but we are evaluating the specimen for the presence of cells. Determination of an organism's clinical significance. So this is again very important, because the number of microorganisms present on the body or inside the body are say 10 to 30. Very large number of organisms present on the body, on the surface and inside the surface. So we want to determine whether this organism is clinically significant or not. So in first lecture, we have discussed regarding the colonizer and contamination and commensal organism. We want to determine whether this organism is responsible for the disease process or not, or actually infection or not. So this is very, very important. So we are applying various definitions to label whether the isolated or identified organism having some clinical significance or not. So this thing we'll discuss later on. Pre-culture information. So for the bacterial examination and for the fungal examination, we are doing culture. So we are examining the specimen. These are the few applications of the microscope in the microbiology lab. So all these applications are different from the applications in the pathology or in physiology or in anatomy that we are using. Now, what are the types of microscopes? These things you already learned in the first MBBS. So I'm not going into detail of these things, but let us see. Hurriedly, simple microscope consists of a single lens or magnifying lens. Compound microscope consists of 2 or more lenses. Compound microscope further divided according to different microscopic method used. Now, this is a photograph of the microscope. There are different parts, mechanical parts, and there are lenses. So, so you read it by yourself. So what are the parts and what are the uses of, what are the functions of these parts? Now, what are the microscopic methods? Now, there are 2, light microscopy and electron microscopy. Light microscopy are further classified into this: bright field microscope, dark field microscope, phase contrast microscope, and fluorescent microscope. So, to the microorganisms nature or characteristic, we are using different types of light microscopy techniques. B is electron microscopy, where there are 2: transmission electron microscopy and scanning electron microscopy, where we are using this microscope for the identification of certain viruses. Is it clear to you, whatever I'm saying? Yes or no? Are you with me? All students are with me? You can write your answer in chat. All students are with me? Okay. यहाँ तक समझ में आया आप लोगों को? Are you getting whatever I am saying? Okay, fine. So, what is the principle of bright field microscopy? All students knows the principle of the bright field microscopy is emitted from light sources, passes through it, iris, diaphragm, and fall on the specimen gathered by objective and magnified image forms, which further magnified by the ocular lenses. This is a simple definition or principle of the bright field microscopy. So this is to be understood. These things we are not asking in the exams, remember. Now, important thing is applications of bright field microscope. Let us see the applications of the bright field microscope. I will show you some photographs or slides, actual slides. And one by one we'll discuss. So application is important for us. Used to observe the morphology of bacteria. I repeat, used to observe the morphology. Now, what is the meaning of morphology? I think all students knows the meaning of morphology, and you already learned in the first MBBS the meaning of morphology. What is the size of the organism? What is the shape of the organism? And what is the arrangement of the organism? Similar way, what is the size, shape, and arrangement of the other cells present in the specimen? Presence of then various organisms like presence of various organisms like bacterial organism or protozoa, or the, I already said in the first lecture when the parasites are divided into. So protozoal parasites are not seen. They are microscopic in size, so they are not seen by unaided eyes. So microscopy is required to identify them, protozoal parasite, and some forms of the metazoal parasite. So metazoal parasite, there are various forms, say egg form or cyst form or different forms. while adult forms are seen by unaided eyes, because the size of the adult form for some is in millimeter to meters. So this form is seen by unaided eye, but the various forms of the parasite are not seen by unaided eye. So for them, we required to use microscopes. Then fungus and algae application is the microscopic examination. Now remember what is clinical application of this? Clinical application is the microscopic examination is a rapid examination. So in laboratory, there are tests we are running. One is rapid test, other are the tests which require some time. Okay. So it is considered as a rapid test. So we'll get a result within 1 hour or within 1 to 2 hours. So this is a very well-known advantage of the microscopic examination. Now let us see the other important thing. Cannot observe microbes less than 0.2 micromillimeter in diameter or thickness, such as spirochetes and viruses. So spirochetes and viruses are under the light microscopic method. So remember, these are the two varieties of the organism. They are not seen. For them, we required to use different type of microscopic method or different types of microscopes. Now let us see the first slide. This is a photograph or slide taken. This is taken from the culture examination. This photograph is taken from the culture examination where colonies are stained. Colonies are taken and staining is done by the gram staining method. Now you see the size, shape and arrangement. We want to see in this slide because other types of cells are not seen. Only one type of cells are seen. So you try to understand size, shape and arrangement. Now size is around 1 to 2 micromillimeter. Okay, I repeat, size of this organism is 1 to 2 micromillimeter. The shape is said cells are circular. Cells are circular. So shape is circular. Size is 1 to 2 millimeter. Size shape is circular, and arrangement, they are arranged in clusters. So you can say a gram now, and then see the Gram's reaction. Gram reaction is, say, cells are violet in color. So these cells are gram positive cocci. Cocci means they are circular. So gram positive cocci arranged in clusters. You can write a statement like this: gram positive cocci arranged in clusters. So in this one statement, all three things are covered. What is the size, what is the shape, and what is the arrangement? So I think this is clear in your mind. Gram positive cocci in clusters are seen because this is a smear prepared from the culture. So that's why other types of cells are not seen. I repeat, because cultures are pure, so other types of samples cells are not seen. Other types means WBCs and RBCs and epithelial cells are not seen because this smear or slide is prepared from the culture. See the next slide. Here I have kept two slides to compare with each other. Now the left side, you see gram cells are violet in color, gram positive cocci in clusters. On other side, gram negative bacilli are seen. Okay, so I'm not saying the arrangement because in the right side arrangement is not fixed. So arrangement यहाँ पे clear नहीं हो रही है. So that's why I'm not saying or writing the arrangement. So if you want to write a report from this, gram positive cocci in clusters are seen. This is this covers the morphology, all 3 points. While in the second side, on the right side, the color is orange or pink. Say gram-negative bacilli are seen. Bacilli means they are rod-shaped. Cocci means they are rounded. So you can write like this: gram-positive cocci in clusters are seen. Second statement: gram-negative bacilli are seen. So here we are speaking important three morphological features along with the Gram's reaction. So actually there are four things. First is Gram reaction, second is size, shape, and arrangement. So we are speaking four things. So when you read a report, when you go to the wards or clinical side and you open up the patient's file and read a report will be like this: gram-positive cocci in clusters are seen. So we may write like this: gram-positive cocci in clusters are seen, suggestive of. Now you write down the name of the organism, what you are suspecting. So what now you say which organism showing such type of morphology. So you think about the organism. You may write like this: gram-positive cocci in clusters are seen, suggestive of Staphylococcus aureus. You can write like this. So you will find report like this in the patient's file. In the second slide, gram-negative bacilli are seen because there are many. There is no fixed arrangement, so we are not writing suggestive of. Okay, so we are only writing gram-negative bacilli are seen. So let us move to the another slide. I think this is clear in your mind. Everybody, yes or no? How to write a report and how to read a report. Is it clear? Okay. Can you able to write and interpret the report when you see a patient's file? I repeat the question. Are you able to write or interpret the report when it is there in the patient's file? Okay, fine. So move to the next slide. Now, let us see this slide. Now here, this is different from the previous slides. In this, there are cells. Now, how to describe the cell? First, speak about the Gram's reaction. So what is a Gram reaction? Anybody can speak or write? What is the Gram's reaction? Violet color cells are seen. What is the Gram's reaction? Write in a chat box. What is the Gram's reaction? Don't worry whether you are right or wrong. Don't worry, write it. Okay, it is not, Rutam answered the question, but it is not gram negative. Remember, we'll teach you Gram's reaction in next practical. So don't worry. Gram positive cocci. Now I am reading the report. Gram positive cocci. Now, what is the next? What is the next thing? Gram positive cocci is a Gram's reaction because cells are violet in color. Gram positive cocci in pairs. Now, gram positive cocci means reaction is the cells are of violet color. They are cocci means they are circular or rounded, and in pairs is the arrangement. So gram positive cocci in pairs, capsulated, because you see around the cell there is a halo, or I think this is clearly seen. So there are two cells, gram positive cocci. They are arranged in pairs because there is a pair of cocci. There is a pair and surrounded by halo. So gram positive cocci in pairs, capsulated, because this halo is of capsule, capsulated. Now, because this smear is prepared from the sputum specimen, so here we are also writing the number. Now number is written like this: few, moderate, and plenty. Now we'll discuss later on what is the meaning of few, what is the meaning of moderate, because numbers are associated with these terms. Say few, 1 to 2 cells, but we are, at this moment we are not discussing this. Here, plenty of gram positive cocci arranged in a pair, capsulated. Now there are other cells are also seen. Now these other cells are WBCs. So along with few WBCs seen. So you can write like this: report gram-positive Chi in pairs capsulated, plenty of gram-positive Chi arranged in pairs capsulated along with few WBCs are seen or few first cells are seen. So you can write a report like this when you see this slide under the microscope. Let us move to the another slide. Now, in this slide, there is a gram reaction. The cells are of pink color. Color is not appreciated nicely, but cells are of pink color or red color or pink color, and there is these cells are of looking like cow cells. Okay, cells are looking like cow cells. So shape is cow. So you may write plenty of gram-negative cow bacilli are seen. So this is a smear prepared from the this is sample. Rice water is sample. Now you see typical cow bacilli are seen. Now this is because sample is rice water in nature. That means patient is suffering from diarrhea. So you can associate this disease and presence of cod bacilli with the cholera. So you can write like this: gram-negative bacilli, cod bacilli, and plenty of gram-negative cod bacilli are seen. Now here you can write suggest you are behavioral quality. Okay. Now you can do further examination and final the report, but within 1 hour you can write this. Let us move to the another staining technique, and that is Jade's staining technique. Now let's discuss staining techniques in an practical. But remember, there is a difference in this staining technique. Both are there is a differentiating staining technique. Differentiating means what? They are differentiating types of cells. That's why they are known as a differentiating staining technique. Okay, so when we are using word differentiating technique, means there are two types of cell differentiated by using two types of dye. So one type of cell take color of one dye, and other type of cells take color of second dye. So if I repeat, we are using this staining technique to differentiate two types of cells, two variety of cells. Okay, by using two types of dye. One type of cell take a color of one dye, while the other type of cell take a color of other dye. So we can differentiate because color of this dye are different. One dye is violet color, other dye is other dye's color is of pink. So this is a concept of the using two dyes to differentiate variety of cells. We will discuss later on in the next practical staining technique. So this is a slide, but this is some specimen where we use a Jade's staining technique. Now remember, the protoplasm organism is acidic in nature. I repeat, protoplasm of the microorganism is acidic in nature. So in microbiology, we are using basic dye. I repeat, in microbiology, we are using basic dye because protoplasm in microorganism is acidic in nature. So acidic protoplasm represents the basic dye. So this is the cells of microbiota. Now microbiota don't take a stain like gram stain. So for them we are using acid staining. So later on we will try to understand what is this. But at this moment, here was prepared from some specimen and brick red color bacilli are seen. So this brick red color bacilli are of microbiota. Now appearance is typical. They are dead in nature. So the beads like structures are bead like structure makes bacilli. Okay. So later on we will see what is the meaning of this. There is a biomorphism also seen. I think you know what is the meaning of biomorphism. So these are the biomorphic dead bacilli, and this is typical of microbiota. Now these microbiota are responsible for the disease tuberculosis. Now there is another smear, another staining technique where bacilli are dead. here typical faded bacilli are seen and pink in reddish pink in color. Now you will see the arrangement of bacilli. So here also we are reading +1, +2, and +3. So later on, we will discuss what is the meaning of this +1, +2, and +3, and what is the meaning of few, moderate, and plenty. Now you see this another scenario prepared from some specimen where reddish pink color bacilli are seen. This is are of microbacteria. You can label it. Suggest your microbacterium tuberculosis. Here in 20, okay. So 25% H2SO4 is a very strong acidic chemical. So the colorizer, no bacteria can withstand against this concentration of H2SO4. When you are using this colorizing agent and bacilli are reddish pink in color, that means this bacilli are absorbing Mycobacterium tuberculosis. So you can label it. Now this is another staining here where we have used 5% H2SO4 as a decolorizing agent. So little lesser than the colorizing agent that we are using for the mycobacteria. Here we are using 5%. So because this is a 5% we are using mycobacterium lapri, showing this. So these cells are of mycobacterium lapri, and we see here robi are seen. Robi means they are arranging like a cigar bundle. Now you try to remember how cigar bundle looks. So cigar they are typically arranged in a cigar bundle appearance. Now this is another senior. This is a general staining technique, and then there is a Y and cigar bundle appearance is also seen. So you can write acid fast organism are suggestive of Mycobacterium lapri, because here we use a 5% concentration of H2SO4 as a decolorizing agent. This is a oocyst. Now you reduce the decolorizing concentration of decolorizing agent, like say 1% to 0.2% H2SO4, then oocyst take this stain or showing of acid fastness. So these are the oocyst of the parasites. So I have given 3 examples. One example is of Mycobacterium tuberculosis, where we have used 25% H2SO4 as a decolorizing agent. In second, I'll show a Mycobacterium lapri, where we have used 5% concentration of H2SO4 as a decolorizing agent. And this is the third, where we have used 0.2 to 1% of H2SO4 concentration. Let us move to the another smear, and that is this smear is taken from the lesions in the throat. Okay, so these are the this is a different type of stain. This is known as the Albert stain. Now Albert stain, we'll discuss later on what is the meaning of Albert stain. Now Albert stain, where there is a safety pin appearance or matchstick appearance, where the organism shows there is a violet color granules seen at the one end of the bacteria, or at the bacilli, or at the two end of the bacteria. So this is a typical of Corynebacterium diphtheria. The organism is responsible for the disease diphtheria. Later on we'll discuss regarding this, but you see the metachromatic granules are seen at the end of the bacilli. Now let us see this slide. Now on right side, same smear was stained with the Gram staining. On the left side, Albert stain was used. So when you use Albert stain, you will find a metachromatic granules. Okay, and typical matchstick appearance. On the other side, the smear is stained by the gram staining. So you see the pattern. The pattern is Chinese letter pattern. You see the typical pattern, but organism are gram positive bacilli. Later on, we'll discuss what is the meaning of this. But at this moment, I want to show you the various type of stains and various type of sites that we are examining under the microscope in the laboratory. Now, move to the unstained preparation. Unstained preparation means wet mount preparation. We are just keeping a sample on the glass slide and keep a and when we'll and on this we are keeping a cover glass or cover slip and directly examine under the microscope. So this is known as a wet mount examinations. Wet mount examination. Now in this slide, you see the hyphal structures are seen. Now here we used a KOH wet mount. So this is for the fungus. Now when we see fungus, they are broadly classified into two categories. There are various classification given for the fungus that we'll discuss in an introduction to fungus. But at this moment, you try to understand fungal infections are classified broadly into two categories. One is fungal infection on the superficial surface of the body, and fungal infection in the organ system. Now, in between there is one category, fungal infection on the mucous membrane. Infection on the superficial surface and mucous membrane. Say superficial surface we are talking, then fungal infection involving the skin, hair and nails, and the mucous membrane like buccal mucosa and I'm just setting something. Fungal infection on the superficial surfaces and mucosa. So here we are taking a scraping or collecting a scraping specimen from the skin, nail and the hair follicle or from the mucosa, and we mix this specimen with the 10% KOH. When we are collecting a specimen like hair and nails, then we are using 40% of KOH. Now this KOH is a digestive reagent. So you may say it digests the keratinized tissues because fungus are inside the keratinized tissues. And when you examine directly, then you will not find fungus. So before that, you have to digest the tissues, keratinized tissues. Once keratinized tissues are digested, fungus is eliminated. So here you see this is a wet mount examination prepared in a 10% KOH. This is another slide where fungal hyphae are seen. This is a scraping taken from the cornea where such type of a picture is seen. Now let us move to the another variety of wet mounts. Another variety of wet mount is wet mount prepared from the stool specimen. Now this is for the parasitic infections. Now you see this is a wet mount prepared from the stool specimen. Now there are two variety of wet mounts are prepared for the stool examination. One is iodine wet mount, other is saline wet mount, where this is prepared from the saline. But when you use iodine, there are two purposes of using iodine. One is it kills the parasite. Second purpose is it gives a color to the parasite. So these are the two purposes to use iodine. So this is a saline wet mount under which the egg of the Ankylostoma duodenale is seen. Now you see this is a typical morphology of the egg form of the Ankylostoma duodenale. Size is around 35 to 40 micromillimeter, oval in shape, and there are blastomeres seen inside. Okay, So 1, 2, 3, and 4 blastomeres are seen. This is a division of the ovum occur, and these are 4 blastomeres. It may be 8, it may be 16. Now this is another weight mount examination where larval forms are seen. Now these forms are microscopic. So these are the larvae of the Ankylostoma duodenale, or it may be a larval form of a Strongyloids stercoralis. So this is a diagnostic. You can write suggestive of this. When it is diagnostic, you can write suggestive of this. This is another weight mount examination where fecal material is also seen in the background. Okay. So when we examine the feces or stool sample under the microscope, there are 2 types of structures. One is parasitic structure, others are the non-parasitic structure. Where parasitic structures are like, say, egg or cyst or worms, or sorry, larval forms are seen, while non-parasitic structures are WBCs, RBCs, epithelial cells, epithelial cells, fungus, hair follicles. So these are the non-parasitic structure. So try to understand when we examine what type of things we are examining under the microscope. So when we examine stool sample, these are the variety of structures seen under the microscope. And here in the center, and photograph is taken from the center portion of the field. And here the 16 blastomeres are seen. Okay, overlapping 16 blastomeres are seen. This is the egg form of Ankylostoma duodenale. Now see, these are the cysts of Entamoeba histolytica or disper, because histolytica and disper cyst cannot be differentiated from the weight mount examination. Left side you see stained with the iodine. So the color of the cyst is different from the weight mount prepared from the saline weight mount. Okay, so I think this is clear. These are the 2 types of eggs seen under the microscope in a stool specimen. Fertilized eggs of the Ascaris lumbricoid on the left side. On the right side, unfertilized eggs of the Ascaris lumbricoid. So later on, we will discuss this when we in this GIT in the month of April or May. Now move to the other type of microscope. Here I completed the bright field microscopic examination. Is it clear to everyone? Application part is clear to everyone? Okay. So move to the next microscopic method, and that is dark field microscopy. Now dark field microscopy concept is very clear. Some structures are not illuminated in a light microscopic examination. Some smaller structures. Say I usually give this example to the students. Say in the night hours, stars are clearly visible or illuminated. Stars are always there in the sky, but in the daytime, stars are not seen because there is a bright field. Okay, in bright field, smaller structures are not seen. But when fields become darker, background become darker, when sky become darker, stars are illuminated. So same concept is utilized over here. When you make a dark background, some structures are illuminated. So we are using the unstained preparation to visualize some smaller structure which are not seen under the light microscopic method. So so this is a very simple concept used in the microbiology. Principal object appears bright against dark background, so our object is illuminated against the dark background. Central opaque area is condenser, block light from the entering the object lens. So here there is a block. Okay, in the central area there is a block. So in previous time, usually we make a dark field microscopy from the simple microscope by keeping 50 paisa coin on the condenser. But now such blocks are available. So you make a dark field microscopy. Or in weight mount examination, you can use dark color dye. So India ink or nigrosin dye is a dark color dye. So when you use this dye, background become darker. So under this darker background, the structures are illuminated or easily seen. So this is a concept. Peripheral annular hollow area allows the light to pass through and focus on the specimen obliquely. So specimen के ऊपर obliquely rays आएंगे, so these structures become illuminated. Reflected light by the specimen enter the objective lens. Unreflected light not enter the objective. Specimen brightly illuminated in the dark background. So this is a concept of the dark field microscopy. Living unstained cells and thin bacteria like spirochetes can be illuminated. So spirochetes are very thinner structure and can be visualized under the dark ground microscopy. Now this, you see this. This is the application. So under the dark background, Treponema pallidum or the organisms, spirochetes are seen under the. So this is otherwise this organism cannot be seen under the microscope in the light under the light microscope. Other method is phase contrast microscope. Now there are phase changes in the light rays induced by the difference in the thickness and refractive index of the different parts of the object. So all cells are reflecting differently. Remember, all cells are reflecting differently. So this difference between the object can be transformed into difference in the brightness of the light intensity. अगर उनकी refractive index और thickness अलग है तो और intensity वो show कर रहे हैं वो भी different होगी. So all cells are differentiated by this way. So this is a application. You can examine the microbial motility, determining the shape of the living cells, detecting the bacterial component, endospore and inclusion body can be clearly visible. Inclusion body is an indirect evidence of the viral infection because viruses are not seen directly under the under this type of microscopes. So for examination of the viruses, we need to use electron microscope. But instead of that, you and this is the indirect evidence of the virally infected cell. So you identify the virally infected cell, not actually viruses, but virally infected cell, and make a diagnosis. This required experience, but one can do this. Fluorescent microscopic method, ultraviolet light. I'm just reading the principle. Ultraviolet light, very short wavelength, not visible to eye, used to illuminate the organism, cells, and particles, which previously stained with the fluorescent dye called fluorochromes. So you can use a fluorescent dye, and this is examined under the microscope, fluorescent microscope. Structures are illuminated differently, give a fluorescent color. Now various dyes are available: acridine, orange, oramine, and fluorescein, isocyanate. Now this I'm not reading or not all these things you know. Now see the slides. Mycobacteria stained by the oramine dye. Now these structures are illuminated. Immunofluorescence of Streptococcus pyogenes. Now you see the organism. They are arranged in chains but illuminated clearly. So gram-positive cocci in chain. Here we cannot use gram-positive, but you can use a word illuminated organism arranged in chains. So most probably they are streptococci. So application, fluorescent microscope. Now this theory you read from the book, and electron microscope. So in our institute we do not have electron microscope, but so for us this is theoretical. So but yes, fluorescent this electron microscopes are available in Vidyanagar in bioscience department, but we do not have because we are not using this. We are using some different technique, indirect evidences for the identification of the microorganism. So actually, there is no need of this at our place. There are scanning electron microscope and transmission electron microscope. So you read this theoretical aspect from the book. So scanning electron microscope, applications of scanning electron microscope, transmission electron microscope, and application of transmission electron microscope. Now, at the end, I'll give you a few questions. You write it down in your notebook, or you may take a photograph of this slide. Or if you want, I may send these slides to you for your reference. Any one student can send me a message on my WhatsApp. I will attach this presentation to that student, and that particular student will send to contact numbers at present. And I'm not a member of your group. So last time one student sent me his mobile number. So I have forwarded presentation to that student, and I hope that student circulated that presentation to whole batch. Yes or no? Everybody received the presentation? Everybody received my last presentation? Okay, fine. So I will send this presentation to you. But I'm reading the questions. Describe working principle of microscope. What are the different types of microscopy and their uses? We have already discussed the uses of microscope or application of microscope in the microbiology laboratory in detail. Enumerate the uses of dark ground microscope, that already done. What is micrometry? This is a first MBBS question, but still you have to write what is micrometer. What is the resolution power of microscope? I skipped this portion because I understand you know these things. What is micron, nanometer, and angstrom unit? I skipped deliberately this portion as well because I hope you know this. What is the advantage of keeping oil when oil immersion lens is used? We have not discussed this because what is the position of condenser and diaphragm when observed through oil immersion objective and why? This is also done in the previous years. Why do we need to stain microorganisms in order to observe them? That we have discussed in detail. What is Brownian movement and true motility? You do not answer this question, 11th question, because you have not discussed till. Later on, we will discuss this. What is the meaning of Brownian movement and what is the meaning of true motility of the microorganism? So up to 10 questions you can answer. 11th you skip. When you join the laboratory or offline classes, you write the questions. We may discuss these questions or clear your doubts when you come to the college. Thank you very much.